Multicellular organisms experience hunger, thirst, sleepiness, excitement, and stress. And all our cells divide to make new cells. But not all of these are things that we consciously will. What is happening is that inside our bodies, communication systems work invisible to us, yet constantly active, relaying information about conditions internal and external to our cells. For example, following a meal, pancreatic cells sense an increase in glucose in the blood and begin making and releasing insulin to stimulate other cells to take it up. Simultaneously within target cells, enzymes mobilize to process the incoming material. Meanwhile, the brain takes note of blood glucose levels and suppresses appetite. And all of this occurs under our radar. As multicellular organisms, we have over 200 types of cells in our body. But when it comes to communication and coordination among these cells, a lot of the responses to environmental changes, whether external or internal, are signaled using what can loosely be called hormones. In animals, hormones originate in a few specialized organs that make up what we refer to as the endocrine system. These glands release hormones only inside the bloodstream. Major endocrine glands include the hypothalamus, pineal gland, pituitary gland, thyroid gland, parathyroid, thymus, adrenals, pancreas, and ovaries or testes. So with this kind of organ-based communication, everyone is called, but only a few cell types do the calling. Human technology using electronic communication networks is only beginning to catch up with the scale and efficiency of how cells communicate with each other. Our cells receive and send signals constantly. You might say they had cell phones long before we got our handheld devices. Some special phones inside my cells are deep inside of me. They manage all my glucose and they act accordingly. If blood should be all glucose flush, it's quickly stored away. And if I need it badly then, adrenaline saves the day. It's like the phone I carry around with just these two exceptions. I never get a monthly bill and always have reception. Cellular communication, as with everything else in biochemistry, depends on specific molecular interactions where the message and the receiver are biomolecules. Binding of a message by the cellular receptor molecule triggers information transmission inwards to the cell and changes its activity. In this lecture, we will see how cells communicate from the role of hormone-based signals created by special cells in complex multicellular organisms to biochemical communications between organisms. We'll eavesdrop on cellular chatter and trace pathways triggered by the fight-or-flight response. We'll also see how errors in communication have profound effects on health. Cellular communications always involve sensing and signaling. It has long been known that even bacteria sense and move toward food sources and away from toxic substances. Even more interesting, bacteria talk to each other, kind of like teenagers at a mall, to find out how many friends are nearby. To do this, bacterial cells use a process called QUORUM sensing, where they release molecules called autoinducers into their environment. The more bacteria there are, more autoinducers are present. If there are many bacterial cells, high levels of autoinducer increasingly bind receptors on their membranes, letting cells know that there are others in the vicinity. What disease-causing bacteria do with this information is fascinating. The leading cause of hospital-acquired infections in the U.S. is the bacterium Staphylococcus aureus. There's a notorious form resistant to antibiotics called MRSA, short for methicillin-resistant Staph aureus. Like military invaders, these Staphylococcus bacteria assess their numbers to determine if there are enough soldiers to successfully pull off a mission. If numbers are too low for an attack, they'll lose to the immune system. But if QUORUM sensing tells them their numbers are sufficiently large, they turn on production of toxins and virulence factors needed for an infection. By doing this, bacteria avoid wasting resources on attacks that have a low probability of success. For humans on this battlefield, snooping on the enemy chatter could prove useful. In fact, scientists are focusing on disrupting bacterial communication. If bacteria are unable to sense the presence of backup troops, it might be possible to prevent dangerous infections. Plants, on the other hand, might seem like they don't need to talk, but they sense and communicate in response to light, gravity, temperature, and water. Inside a plant, messages carry information for specific cells. In the fall, cells in leaves get directed to break down chlorophyll, exposing red and orange pigments and to weaken connections between the leaves and the plant, so it drops its leaves. The color changes we admire in trees are occasioned by plants talking to themselves. More surprising are some of the other tricks plants have up their leaves. Some plants recognize family members, that is, genetically related individuals. Such plants share resources with relatives by limiting the amount of soil nutrients they take up. If they sense a stranger, though, they compete by putting out extra roots to take up more nutrients. Molecules exuded from roots may help distinguishing strangers from family. We learned in the lecture on lipids how plants respond to insect attacks by releasing volatile organic molecule signals, such as methyl jasmonate, into the air around them. This alerts other plants to predators. The signals trigger increased production of chemicals like polyphenols that affect the taste of the plant, or proteinase inhibitors that inhibit digestion of the plant by the predator. Such knowledge may help us develop ways to protect crops. What if farmers sprayed signals like methyl jasmonate instead of pesticides over their fields? Plants could defend themselves by preemptively releasing molecules to protect themselves against attack by insect pests. Not surprisingly, the most complex communication systems are found in multicellular animals. Many of the signaling molecules come from the endocrine system and go by the general name of hormones. The term hormone means different things to different people, so as I talk about them here, hormones are simply molecules that travel in an organism to communicate a message. We can divide hormones into two broad groups. One, molecules that act within minutes through binding to receptors on the surface of cell membranes, and two, steroid hormones that act more slowly and bind to receptors inside of cells. Many hormones are relatively small proteins. Insulin, the hormone that controls blood sugar levels, for example, has only 51 amino acids. A quick-acting group of hormones called catecholamines is produced by the adrenal glands on top of the kidneys. Norepinephrine and epinephrine, also known as adrenaline, have the same ring structure called catechol attached to an amine, hence catecholamine structure. Slower-acting steroid hormones such as estrogen, testosterone, and cortisol are created from cholesterol, as we learned in Lecture 16. These signaling molecules travel inside the cell membrane to reach their receptors and tend to act over time periods of hours or days. Another diverse set of signaling molecules to note are the cytokines. They bind on the surface of cell membranes, similar to quick-acting hormones, and are especially important in the immune system. How all these signals get to their target cells is equally varied. Signaling in animals that uses the circulatory system is called endocrine signaling. Signaling that only involves neighboring cells is called paracrine signaling. A signal molecule that acts only on the outer surface of the same cell where a signal was created is called autocrine. An intracellular signal molecule that stays and signals inside the membrane of the same cell is called intracrine. There are also exocrine glands that release outside the body, such as sweat glands that send signals to communicate with other organisms. Cells receive a signal starting when the signal molecule binds to a receptor protein, often located on a cell's membrane. But for steroid hormones and also thyroid hormones, the receptor is located inside the cell, so such signals must cross the membrane and enter the cell to meet up with their receptors. For intracellular receptors involved in steroid signaling, a typical outcome is that cells adjust the amount and/or the timing of the synthesis of specific proteins. For signaling occurring through membrane receptors, the receptor protein conveys the message through a chain of other cellular molecules into the interior of the cell. In some types of membrane receptor signaling, enzyme activities are altered. Affected enzymes include protein kinases, which phosphorylate target proteins, and phosphatases, which remove phosphates. As we saw in the metabolic control lecture, adding and removing phosphates is an easy way to control whether cellular proteins are active or not. A good example of such signaling is the fight or flight response that evolved to prepare an organism in a dangerous situation for fighting with or fleeing from predators or competitors. This same stress response system kicks in when we get cut off in traffic by an aggressive driver, interview for a job, or even simply make a presentation before the boss. Any response to stress takes energy as well as a toll. First, the energy. This involves mobilizing stores of food to provide additional glucose. It begins in a section of the brain called the amygdala, which is involved in memory, decision-making, and emotions such as fear, aggression, and anxiousness. When the brain perceives danger, it sends messages to the adrenal glands above the kidneys, stimulating them to secrete hormones: epinephrine, also known as adrenaline, norepinephrine, and cortisol. Norepinephrine is also produced at low levels normally and acts similarly to adrenaline, so we'll focus on adrenaline first and then take a quick look at cortisol. Adrenaline is familiar to most people and is often credited with giving people extraordinary strength or endurance under stress. Here's how it works at the molecular level. There are quite a few steps in the pathway from adrenaline binding at the cell surface to the final cellular changes, just as a directive from the CEO has to go through many levels in the organization before reaching the people who implement the decision on the factory floor. There are three important themes. First, the signal arriving at a cell must be bound by a receptor for any action to follow. Second, the receptor passes the message to a chain of molecules, forwarding the memo, till finally a kinase or phosphatase is activated. Third, the relevant enzyme either phosphorylates or dephosphorylates target proteins and changes their activity. Okay, let's take a closer look. Cells in the liver are primary targets of adrenaline signaling. Here, adrenaline binds to a receptor called the beta-adrenergic receptor on the cell surface. The beta-adrenergic receptor has a structure of 7 helices called 7-TM, short for 7 transmembrane domains. Like a mountain road with switchbacks, the polypeptide chain of the beta-adrenergic receptor folds back and forth through the cell membrane 7 times. When adrenaline binds the beta-adrenergic receptor, the receptor changes shape subtly. This change allows the receptor's tail inside the cell to interact with another protein called a G protein. So the receptor is called a G protein coupled receptor. The G protein is the first in a series of minions that transmit the message through the cell. Now, G proteins are a class of proteins that bind to guanine nucleotides like GTP and GDP. There are many different G proteins, each specific to slightly different signaling pathways. Each G protein is actually made up of 3 polypeptides called alpha, beta, and gamma. In the absence of a signal, the alpha part of the G protein has a GDP bound to it. And in this state, all 3 polypeptides hang together tightly, snoozing until prompted by the receptor. Binding of adrenaline causes the receptor to interact with and alter the conformation of a G protein in the cytoplasm of the cell. When nudged by the tail of the receptor, the G protein changes shape slightly. The alpha unit lets go of its GDP and picks up a GTP in its place. The alpha subunit bound to GTP then separates from beta and gamma. It's the freed alpha subunit bound to GTP that passes on the message to an enzyme called adenylate cyclase. The alpha subunit activates adenylate cyclase, which in turn makes a modified nucleotide called cyclic AMP or CAMP. Lots and lots of it. Unlike other nucleotide forms, cyclic AMP doesn't have any functions for energy or for making nucleic acids. It's simply a messenger. So what does a small diffusible messenger like cyclic AMP do to implement what adrenaline wanted to do? Well, it turns out cyclic AMP can bind to many different proteins and cause many different downstream effects. But in this situation, we'll follow cyclic AMP as it binds to an enzyme called protein kinase A, or PKA. Remember, signaling involves passing the signal that arrived at the cell through a series of molecules, often resulting in activation of kinases or phosphatase. PKA is the first of these kinases. Now, like all kinases, PKA catalyzes addition of phosphate groups to other molecules. What other molecules? Well, the name protein kinase A tells you that it adds phosphates to other proteins, thus affecting whether or not they are active. So let's take a step back and think about what the organism needs in a stressful situation. The number one need is energy in the form of glucose. From what you've learned already, you know that glycogen, a polymer made of glucose, can be broken down to obtain glucose. You've also learned that glucose can be made in the liver by the process of gluconeogenesis. So if cells need glucose, it would be likely a good idea for them to activate key enzymes for both glycogen breakdown and glucose synthesis. What does PKA do to achieve these goals? Well, let's start with the breakdown of glycogen, for which the enzyme glycogen phosphorylase must be activated. One way to activate it involves phosphorylating the enzyme. PKA doesn't directly phosphorylate the enzyme, but it does the next best thing. It puts a phosphate onto another enzyme called phosphorylase kinase, delegating it the task of adding a phosphate to activate glycogen phosphorylase. Active glycogen phosphorylase can then break down glycogen to provide glucose compounds. And as you already learned in the lecture on metabolic control, when glycogen is being broken down, glycogen synthesis is turned off. So phosphorylation by PKA shuts down the enzyme for making glycogen, known as glycogen synthase. As a result, glycogen is not being made and broken down simultaneously. Okay, we've finally reached the end of one signaling pathway that started in the amygdala of the brain and ended with the activation of a glycogen phosphorylase to release glucose units from glycogen. If you've been counting, that's 10 steps. So why so many steps? Don't we want glucose in a hurry? Why not have the signal activate glycogen phosphorylase directly? Well, the answer to that question lies in the amplification of the signal that occurs with multiple steps. Like a chain letter where each recipient sends the letter on to 7 people, each of whom send it to 7 more, each step in the signaling pathway has the potential to boost the signal. It's like mobilizing a biochemical smart mob or volunteer fire department. Within minutes of perceived threats, your bloodstream is awash with glucose. It also favors the synthesis of glucose in the liver. And it does this by stimulating breakdown of a molecule called F26BP. F26BP acts to inhibit glucose synthesis by inactivating a key enzyme known as F16BPase. And it simultaneously favors glucose breakdown by activating the glycolysis enzyme known as PFK. So when F26BP is present, glycolysis runs and gluconeogenesis is inhibited. When that activator gets destroyed, though, as it is when PKA is active, glycolysis is inhibited and gluconeogenesis is activated. So as a result, glucose is made by the liver through gluconeogenesis. So we've seen that as a result of adrenaline signaling, a series of reactions were set in motion, culminating in providing plenty of glucose to the body, both by breakdown of glycogen and new synthesis of glucose in the liver. Of course, for every on switch, there must be an off switch, because the enzymes we've discussed here were activated by addition of a phosphate. The off switch involves the removal of the phosphate that was added by pKa. An enzyme known as protein phosphatase removes phosphates from all the enzymes I've described here, and when there is an excess of glucose. And what activates protein phosphatase? Insulin. Yes, insulin opposes the actions of adrenaline. This is completely consistent with insulin's role in stimulating cells to take up glucose when blood levels of glucose rise. When there's plenty of glucose in cells, it's time to shut down pathways that produce more. There are two other off switches for glucose I want to mention before moving on. They both relate to activation steps earlier in the pathways we just traced. The first involves the G protein alpha subunit that was split off and activated when GTP bound to it. If that activation isn't stopped in some way, the pathway for glucose production will continue even after the cells have plenty of glucose. So how is it turned off? Well, it turns out that G protein alpha subunits actually gradually catalyze conversion of their bound GTP back to GDP. This inactivates the alpha subunit and allows the beta gamma subunits to rebind with the alpha. This entire rebuilt complex bound to GDP then returns to the beta-adrenergic receptor to await the next signal. The other signal that needs to be turned off is the small diffusible cyclic AMP molecule. If high levels of cyclic AMP were allowed to persist in the cell, pKa would be left in the active state and phosphorylation of enzymes would continue. To prevent this, cells have enzymes called phosphodiesterases that break a bond in the ring of cyclic AMP, converting it to AMP, and that turns off the signal. Okay, so we followed the events that are set in motion by the binding of adrenaline to its receptor on liver cells, and the resulting flood of glucose in the bloodstream to help fuel the response. In addition to the short-term stress response for energy production by adrenaline, a longer-term response to stress comes with the production of cortisol. Now, cortisol is a steroid hormone that is a member of the class known as glucocorticoids. Cortisol's effects overlap somewhat with those of adrenaline. But in addition, cortisol keeps the system on high alert over extended periods of time. Normally, once adrenaline has done its thing, the system returns to a no-danger state. But if the threat is perceived to be ongoing, cortisol keeps the stress pedal depressed. Like other steroid hormones, cortisol exerts its effects by binding to a steroid hormone receptor inside of cells. When a steroid binds, the steroid hormone receptor acts to affect the synthesis of specific proteins. This is a route of communication that is relatively slow and may require hours to fully activate. Among the proteins whose production is increased by cortisol are the enzymes used in gluconeogenesis. To obtain even more glucose, cortisol stimulates fat and muscle breakdown to provide fatty acids and amino acids that can be funneled into glucose synthesis. In addition, cortisol can suppress activity of the immune system and attempt to divert all resources for dealing with immediate threats. These actions of cortisol are useful in combining to provide energy to an organism that needs to get its brain and its body geared for action in the face of continued danger. The downside is that low levels of chronic stress, like worrying about being able to pay your bills, also triggers the cortisol response. In addition to elevating your existing blood glucose levels, even when you aren't literally going to fight or flee, cortisol also stimulates your appetite so that you load up with calories even more. Stress eating and chronically elevated blood glucose are not recipes for good health. Over time, increased blood sugar and chronically elevated appetite, coupled with loss of lean muscle mass, can lead to obesity and insulin resistance. Now, paradoxically, there's a partial upside. Cortisol's suppression of the immune system can be exploited in treating diseases related to inflammation and overactivity of the immune response, such as occurs in rheumatoid arthritis and allergies, as well as eczema. Long-term use of glucocorticoids, though, does have potential negative effects, such as loss of bone density, weight gain, cataracts, and glaucoma. Research into drugs that could deliver the benefits without the side effects is ongoing. Another signaling molecule involved in the brain's response to stress is norepinephrine, which also functions to maintain alertness, readiness for action, and normal levels of respiration. Okay, so we've seen that cellular signaling can be surprisingly complicated. Now I'd like to turn our attention to another category of signaling in which external signals can induce changes in cells that tell them to divide. Like the beta-adrenergic pathway, this one also relies on receptor binding, relaying of the message through various go-betweens, and finally activating kinases that bring about changes in the cell. A classic example of such a signaling system involves a hormone called epidermal growth factor, or EGF. One of the functions of EGF is to stimulate cells to divide. Epidermal growth factor is a polypeptide containing 53 amino acids, and it binds to a cellular receptor known as the epidermal growth factor receptor, or EGFR. EGFRs belong in a class of receptors called receptor tyrosine kinases. The name derives from the fact that they have a latent kinase activity. Kinases, as you know, add phosphates, and the EGF receptors specifically add phosphates onto tyrosines in proteins, hence the name tyrosine kinases. There is an entire family of proteins, each slightly different from the other, that can serve as receptors for EGF and related peptide signals. All the receptors are transmembrane proteins anchored in the cell membrane. One end of the receptor peptide sticks out of the cell, while the other end extends into the cytoplasm. Receptors for EGF work in pairs, with two of them needed to pass on the message when EGF binds to them. Before they encounter EGF, the receptors exist as monomers. When EGF shows up, the two monomers move closer together, drawn together by their shared affinity for EGF. When they get close enough, the kinase activity in the tails of the receptor monomers gets stimulated. And what do they phosphorylate? Each receptor tail phosphorylates its partner on tyrosines in its amino acid sequence. The phosphorylated tyrosines on the EGF receptors act as beacons, attracting other proteins in the signaling pathway located in the cytoplasm. Several different proteins, upon interacting with the phosphotyrosines, then activate other enzymes that pass on epidermal growth factor's message. One enzyme pathway that is very important in stimulating cells to divide is known as the MAP kinase pathway. It involves numerous intracellular protein messengers, but the one we will focus on is a protein called RAS. RAS is a lot like the alpha subunit of the G protein in the beta-adrenergic pathway. It is inactive when carrying GDP, but when it binds GTP, it is activated and stimulates the rest of the pathway. The RAS signaling pathway involves a kinase cascade like we saw with epinephrine signaling, but there are more kinases involved. The last kinase in the cascade is known as MAP kinase, which is short for mitogen activated protein kinase, where a mitogen is a mitosis generator, something that induces cell division. It is this MAP kinase that brings about the changes that lead to cell division by phosphorylating a slew of target proteins. So binding of epidermal growth factor to its receptor results in activation of target proteins by MAP kinase that causes the cells to divide. The interesting aspect of EGF signaling isn't how the pathway gets activated, but instead how it gets turned off. This is important because uncontrolled cell division is cancer. So what's the off switch? Well, there is more than one off switch, but an important one is found at the RAS step. Like the G protein alpha subunit, RAS slowly hydrolyzes GTP to GDP, thus inactivating itself over time. Mutations that destroy RAS's ability to hydrolyze GTP leave it perpetually in the on state. And when that happens, cells continue to receive the message to divide even when they shouldn't. And if cells divide when they shouldn't, I think you know what that means. Cellular signaling is not unlike the children's game called telephone. A message starting with one child gets whispered sequentially to a series of children. At the end, kids discover how different the last message received was from the message at the start. Now, fortunately, the cells in our body communicate a bit more reliably than children do. Unfortunately, when cellular whispers are misunderstood, the consequences are much greater. In the next lecture, we'll no longer be floating our cellular signals through the bloodstream. Instead, we'll get rid of all speed limits and turn to signaling via nerve cells. With some of the same molecules we saw here as hormones, we'll see that neurotransmitters are blazingly faster than any other form of communication in the body.